So some of you were asking me, no, my languages are English, Bengali, and Hindi. No Tamil, sorry. Okay, so some of you were asking me in the last session that whether you can get the YouTube links. I had shared the links, the YouTube links, as well as the links to the presentations in the forum. If you need, you can check it.
Hello, ma'am.
Yes, hello.
Ma'am, YouTube links once more send me, ma'am.
Yes, I had actually sent a mail in the forum where the YouTube links were there. So if you want, I can send it again also. No problem.
Okay, ma'am.
Okay, so let's begin today's session. So in today's session, we will be mainly discussing about the diversity of the antibodies. So in last session we were discussing about the structure and of the B cell receptors and the T cell receptors, how the B cells develop and they mature and differentiate, how the T cells develop and differentiate. And in today's class we will actually go through briefly through the structure of antibody and how the diversity of antibody, that is, we know that antibodies can actually potentially recognize all types of chemicals, not only proteins but all types of chemicals it can actually recognize. So we will actually go through today how this kind of diversity of the interaction repertoire of antibodies are generated. So first let's go through briefly what is the structure of an antibody. I hope you are also quite clear with that. So an antibody contains the light chain, one light chain and one heavy chain, right? What is the light chain here? This is the light chain, and this one is the heavy chain. So therefore in a typical antibody there are how many light chains and how many heavy chains? There are 2 light chains and 2 heavy chains, right? So this light chain and heavy chain, how are they different? First of all, these light chain and heavy chains, they have 2 distinct regions. One is the variable region and another is the constant region. So this top portion, you can see whatever is shown here in red, this is the variable region, and the regions and the domains here shown in blue are actually the constant regions. And you can see in the light chain there is one variable region and one constant region, whereas in the heavy chain there is one variable region and 3 constant regions, right? And how are these variable? Yes, go ahead.
Ma'am.
Yes, please go ahead.
Ma'am, your voice breaking, ma'am.
Your voice is not very clear to me.
Yeah, ma'am.
Can you tell me again, or can you write in the chat if required? Okay. So what is the difference between this variable region and the constant region? The variable region is the region which is actually responsible for binding to the antigen, right? So the variable region is the antigen binding region. And in last session we were discussing about some of the effector functions of the antibodies, right? The neutralization, opsonization, these kind of effector functions, these are usually brought about by the constant region. And these two chains, the heavy chains and the light chains, they are joined by disulfide linkage, right? So this is a typical structure of the variable region of the antibody, the variable region and the constant region. Now the variable region actually has some hypervariable regions as well. Hypervariable regions. So what does it mean? First of all, the variable region is the region which actually varies. That is why it has its name like variable, right? Varies means what? So it can show different types of, there are possibilities of generation of various types of variable region which can actually potentially recognize several types of antigen, right? And in between these variable regions, there are some regions which are responsible for directly binding to the antigen, right, which are called the hypervariable region or the CDR. So here we will actually look at that kind of a structure. So this is actually the variable region of the light chain. So all my figures are taken from Janeway for your reference, Janeway's immunobiology book. Okay, so this is the typical diagram of a variable region of the immunoglobulin light chain. And here you can see that these variable regions are mainly composed of beta sheets. So these are the beta sheets you can actually see. So there are many beta sheets. There are usually anybody else facing any problem from my audio? Is my audio clear? Okay. Okay. I think my audio is clear. If anybody is having any problem, please you can check your network once again. Okay, thank you. Alright, so these are the beta sheets which form the variable regions or the immunoglobulin fold, the so-called IG fold. And these beta sheets, you can see here that these beta sheets has some of the loop regions, right? So these are the few loop regions that are termed here as CDR, right? CDR is nothing but complementarity determining region. So complementarity determining region is nothing but the hypervariable regions. That means the regions which go, which actually undergo various, various variations. That means there is much diversity in these regions which are principally associated with antigen recognition, right? So these are the CDR regions. And the interesting thing here is that we will in the next slide see that how these variable regions are actually encoded in our genome, right? So these are all proteins and they must be encoded in our genome. And the light chain and heavy chain, they are encoded in this way. So the light chain has in the gene it has 3 segments. One is the V segment, another is J segment, another is C segment. So V stands for variable, J stands for joining, and C stands for constant, right? So here I would just like to tell you one thing, that the variable region of the light chain and the heavy chains, they are encoded by V, J and D. D segments. So popularly known as V, D, J. These are the three segments of the gene which encode the variable region of the light chain and the heavy chain. Now interestingly, there is no D segment here in the light chain. You can see that there is no D segment here in the light chain, whereas in case of heavy chain there is a D segment, right? So the variable region of the heavy chain is encoded by the VDJ region, whereas the VJ segment determines the variable region or encodes the variable region of the light chain of the antibody, right? And interestingly, why am I saying that how these antibodies are very diverse? Because there are actually many V, J and D segments in our genome, right? So in the genes there are several V segments as well as several J segments and several D segments. So that means when a particular V segment pairs up with a particular J segment, it creates a particular type of antibody which is very unique than when another V segment pairs with another J segment. I think you are being able to understand this particular concept. That is, suppose V1 pairs with J1, then it creates, suppose, antibody number 1, right? And if V2 pairs with J1, it will create antibody number, it will be antibody number 2. If V3 pairs with J1 or J2, it will be, suppose, antibody number 3, like this. So you can understand there are actually several combinations of these gene segments that contribute to the diverse antibody repertoire that is present in our body. Now the interesting thing is, how does these several combinations arise? The answer lies in somatic recombination. So here it is mentioned, you can see that these VDJ segments, these all segments are there in our germline DNA, right, which undergoes somatic recombination. So in our body cells, that is in our B cells, these undergo recombination, and this recombination takes place in a particular process. I will just go through it also. But this somatic recombination actually allows the rearrangement of several V, D and J segments. So just to give you some details of this particular thing, here just take a look at this gene segment. Here you can see there is first L. L stands for what? L stands for leader peptide. So we know that an antibody is an extracellular protein, right? So it is secreted also from the cell, from the B cells or from the plasma cells. So if it has to be secreted, then it has to go through the secretory pathways of the cell, right? And in that case, the leader peptide actually helps it to go through the secretory pathways. So that is why there is an L segment which actually codes for the leader peptide. Then comes the V segment. I already talked about variable segment, D and J segment. And the C actually will encode the constant region. Okay, so in the first round of, and this is for the heavy chain. I already told you that in the light chain there is no D segment, only V and J segments are present in the light chain. Okay, so first, in the first round of recombination, this is recombination round 1, the D and J segments, particular D and J segments recombine to form the DJ segment, right? And in this next round of recombination, that is recombination round 2, the V segment, one particular V segment recombines with the DJ segment to form the VDJ recombined gene segment. So these are the two rounds of recombination that takes place while developing the heavy chain of the antibody. Now what happens? Now the next steps you can see here, the VDJ, all the segments are actually formed here. Now this is actually transcribed. So this is transcription, and this is the primary mRNA. How do I say this is a primary mRNA? Because you can see a poly A tail is present here. Alright, so this is the primary mRNA, and then as we all know that primary mRNA undergoes a particular event to form the mature mRNA. What is it? Can someone tell me? Splicing, exactly. So this is the event of splicing that takes place here. Yes. Yes, Khushi, don't worry. I will actually go through some of the practice questions. Don't worry. Okay, so the primary mRNA undergoes splicing to form the mature mRNA. And in here you can see that these intron regions, so these are the intron regions. Here also there is one intron region, and the intron regions in between the constant regions. So these are all the exons, right? And the introns are deleted in this splicing event, right? And this forms the mature mRNA for the heavy chain, and which actually codes for, you can see these blue segments codes for the constant region, CH1, CH2 and CH3, right? 3 constant regions. And this one is VDJ. This segment actually codes for the variable region. Now the interesting thing is that I was talking to you about these CDRs, right? I was talking to you about these CDRs, and these CDRs are actually encoded by the VDJ segments, and the CDR1 and CDR2, they are actually coded by V segments, whereas CDR3 is the most interesting and the most important one. Why? Because CDR3 is a particular loop of this loop is actually coded by the 3 segments, the V, D and J. So it is kind of a junction. So suppose it is like this. So V, D and J, all these 3 segments at a junction form a junction to encode the CDR3, right? And that is why CDR3, we will see later on also that CDR3 is the most diverse loop or the most diverse hypervariable region of this light chain or heavy chains, right? So this is how it undergoes genetic gene recombination, right? And then finally, by splicing, the heavy chains and the light chains, the mature heavy chains and the light chain mRNAs are produced, and which finally encodes for the polypeptides. Alright, now let's look a bit more into the recombination process. Now this recombination process actually takes place because of presence of a particular signal sequence, which is called a recombination signal sequence, RSS, recombination signal sequence. So what is the necessity of these recombination signal sequences? So they actually determine which V segment can pair up with the J segment, or which D segment can recombine with the J segment, like that. So it helps in the recombination of the VDJ segments. So how does it look like? So this recombination signal sequence, there are two types of recombination signal sequences. One is 23, another is 12. What is this 23 and 12? So basically you can see that there is either a 23 base pair region or a 12 base pair region. Here this is the 23 base pair region and this is the 12 base pair region, which are flanked on both sides by heptamer and nonamer sequences. Here also you will find the same thing. There is a nonamer sequence and there is a heptamer sequence, flanking the both sides of the 23 base pair or the 12 base pair spacer, right? And there is a rule in this recombination signal sequence, that is, our 23 base pair actually, or our 12 base pair actually pairs up with a 23 base pair recombination signal sequence, while this recombination, genetic recombination, takes place. So this is actually a 12-23 rule. 12-23. So a 12 base pair recombination signal sequence will always pair up with a 23 base pair signal sequence, and not 12-12, 23-23. This kind of recombinations does not take place, right? So only 12-23 is a potential recombination event. And you can see here that these V segments and the J segments and the D segments as well, they are flanked by this kind of recombination signal sequences. Here you can see that this is a 23 base pair sequence, whereas this is a 12 base pair sequence flanking the ends of the V segment or the J segments or the D segments as well. Now what happens that this 12 and 23 base pair recombination sequences, they combine. They actually, what happens is that so suppose there are two sequences like this, V1 suppose and J1, and what happens is that there is, suppose this is a 23 base pair sequence and this is a 12 base pair sequence, and they have their transcriptional orientation in the same orientation, right? So that means they will be transcribed in the same orientation. Then what happens, so there's a kind of loop that forms. So that this recombination event can take place and the intervening loop can actually be excluded. So suppose V1 is pairing up with J1 in this case. This is how the recombination takes place. Now there is one more important event which takes place here. In case of CDR3, I told you already that CDR3 is the loop which is actually encoded by V, D and J segments, right? And it forms a junction between the V, D and J segments. And this is why the CDR3 also exhibits a junctional diversity. What is that? So when these V, D and J segments, when they recombine to form the CDR3, there are certain events which take place which are not recombination but some addition of certain nucleotides, random addition of certain nucleotides, which also CDR3 is present in the heavy chain as well as the light chain. So this, if you go through this diagram once again, CDR3, this kind of variable region is present in both the light chain and the heavy chain. If the CDR3 contains segments of both V, D and of all V, D and J segments are present in the CDR3, that means it is a heavy chain, is part of heavy chain. If the CDR3 is part of a light chain, then it will be formed only of V and J segments, because there is no D segment in the light chain. Okay, so this junctional diversity is one concept which is actually random addition of some nucleotides. So these are known as P nucleotides and N nucleotides. So what are these P and N nucleotides? They are basically, so P stands for palindrome, and N stands for non-template coded. So what are these nucleotides? So these nucleotides are added very randomly, and you can understand if some nucleotides are being added randomly, then it will create more and more chances of creating an antibody molecule which is more and more specific towards the antigen, right? So therefore the junctional diversity is actually brought about by the addition of certain of these types of nucleotides. So how is this possible? Let's see. When the so here it is shown when the D and J segments are recombining. You can see the D and J segments are undergoing recombination, and these are the RSS. These two are the RSS, right? These are the D and J segments, and these are the RSS which are brought together, and these RSS will also be 12-23, will also follow the 12-23 rule, right? And when these RSS, the recombination signal sequences are brought together, the first thing that the protein which is important, which actually is crucial for this kind of recombination event, are RAG1, RAG2. So these two proteins form dimer, RAG1, RAG2 dimer, and which is the protein which brings about this kind of recombination event. So when these RSS are brought together, the RAG1 and RAG2, they recognize these RSS, right? So the importance of the RSS is that it is being recognized by these RAG1 and RAG2 proteins, and what happens is that these RAG1 and RAG2 proteins, they cleave at a certain point. Where is the point? They cleave the after the nucleotide, after the last nucleotide of the D or J segment, right here. Okay, here you can also see these two arrows. They point out the location where the RAG proteins will actually make a cleavage on the gene, on the DNA, right? And when this RAG complex cleaves at this particular positions, they actually generate DNA hairpins. So here you can see that this kind of DNA hairpins will be generated. All right, and then there is actually a cascade of events that take place, and here the Artemis and DNA PK complex, so DNA protein kinase complex along with the Artemis complex, it actually helps to open the DNA hairpins. So when this opening of DNA hairpin takes place, see here, these are the points where the DNA hairpins are being opened, and the result you can see here. When the DNA hairpins are opened, so this is kind of a, suppose TC and AG, they were bonded, right? They were forming this DNA hairpin, right? And when you cleave at this point, when the Artemis DNA PK complex cleaves at this particular point, then what happens, this actually opens up and goes TCAG, sorry, TCGA, right? So this is what you can see, this TC and AG, right? They are complementary nucleotides, right? So they actually come together on the same strand. So here you can see that how the DNA hairpin will be cleaved. Depending on that, some nucleotides are being added to the D segment at the junction of the D and J segment. Here also you can see that AT, they come on the same strand. So they are now, these nucleotides are now added on the same strand after the J segment. So when these D and J segments will recombine, there will be, these nucleotides will be added in between, in the junction between D and J segments. So that will create a diversity, that will create a possibility of efficient antigen recognition, right? So these are the palindromes actually. So these are the P nucleotides, because you can see that TCGA, this will form a palindrome, and that is why these are the P nucleotides. These are called the P nucleotides. Then further what happens is that some random nucleotides are next added. Okay, so this is performed by the enzyme TDT, which is called the terminal deoxynucleotide transferase. This is the enzyme called TDT. What does it do? It actually, so these ends were generated here, right? After the action of the Artemis PK complex. And after these ends, the TDT enzyme actually keeps on adding some more nucleotides. Here you can see the nucleotides that are added by the TDT enzyme are shown in these boxes. Okay, so these are some additional nucleotides which are added by this TDT enzyme. Okay, and what is the that is why this is called N nucleotide, because there is no template. It is not added according to any template. This is random. This is random addition. So that is why this N nucleotide or non-template coded nucleotides which are added by TDT. Now what happens? These strands are now paired. These strands will now try to pair with each other, and you will see that there will be some nucleotides which will having mismatch. So here you can see that this A and these T, they are having some mismatches. So these terminal nucleotides which are having some mismatches are actually removed by an exonuclease. So these terminal nucleotides are now removed by the exonuclease. And finally what happens is that this gap, so there is a gap here and there is another gap here. So this gap is now filled by DNA synthesis, by DNA polymerase. So the complementary nucleotides are now synthesized by using DNA polymerase, and then there is a complex of DNA ligase and XRCC which actually now ligates these points, these gaps, right? So these gaps are now filled by the DNA polymerase and ligated by the DNA ligase and XRCC complex, right? So now you can see here, so these are the P nucleotides which are added. P, why? Because they are palindromic, so that is why they are P. These are the random nucleotides which are non-template coded, which are N nucleotides, and then again at this end also there are some P nucleotides. Now every time this process will take place, every time this recombination will take place, different nucleotides will be joined as P nucleotides and N nucleotides. So you can understand the number of times these recombination events are taking place. Several, like numerous combinations of P and N nucleotides are being added, which is actually adding to the diversity of the antibody repertoire, right? So this is one very important event which is taking place, and this is responsible for the junctional diversity of the CDR3 region, right? So we are actually going through some of the events which actually help to um, diversify the antibody repertoire of our body. The first one was the recombination of various V and D and J segments. The second one was junctional diversity, right? So the first one was combinatorial diversity, which is the recombination event. This is the junctional diversity. And later on we will also study about, in the later sessions, we will study about the somatic hypermutation, which actually takes place on the, so these are certain mutation events which take place on the variable regions. So the variable regions are already encoded by this kind of recombination event and the junctional diversity events, but finally upon those variable regions again some mutations again take place, which actually are known as somatic hypermutation. So we will study that later on. And also one more diversity of the antibodies is provided by several antibody classes, which are actually coded by the or which are actually determined by the constant region of the antibody, right? So here you can see in the mouse and the human genome there are several constant regions. Here you can see C mu, C delta, C gamma, C epsilon, C alpha. These are several regions, these are several segments of the constant region also, which are present in the gene of the constant region. So these are the several antibody, these actually code for several antibody classes. What are the 5 classes of antibodies? IgM, IgT, IgG, IgE and IgA. And this is how the structure actually looks like, and you will find out that there are several differences between them actually. So IgD and IgM, you can see that in case of IgD there are 3 constant regions, right? 1, 2, 3. Whereas in case of IgM you can find 4 constant regions. And these antibodies also form several like pentameric, dimeric, tetrameric forms. So in that case also they are different, and they have also different effector functions. So they demonstrate different effector functions. And here also in case of IgE also you will find 4 constant regions, right? So IgE is actually encoded by C epsilon, the regions C epsilon. C delta codes for IgD antibody class, like that. So these way the antibody, several antibody classes are also observed. Alright, so I hope this is more or less clear with all of you. If you have any questions you can ask me, or we will go through the practice questions. Okay, so if you have no more questions, so we will go through the questions, solving the questions. Okay, this is the first question. Introduction of P nucleotides during immunoglobulin gene rearrangement is also known as VDJ diversity, combinatorial VDJ diversity, diversity caused by somatic hypermutation, junctional diversity, combinatorial light chain and heavy chain diversity. Please post your answers in the chat box. Okay, most of you are actually giving correct answer. That is C, which is junctional diversity, right? So I already mentioned that P nucleotides and the N nucleotides, these are introduced at the junctions of the V, D and J segments, and this kind of diversity is known as the junctional diversity. Alright, which is different from the combinatorial VDJ diversity, which is actually brought about by the genetic recombination, thus recombination events, which are aided by RAG1, RAG2 enzymes, right? So this is genetic recombination, which brings about combinatorial diversity, and the junctional diversity is actually brought about by the P and N nucleotide additions. Somatic hypermutation also I talked about. This is mutations in the variable regions, which actually help to diversify the repertoire. So that is it. This is the answer. Let's go to the next question. The exon for the CDR3 loop of the heavy chain is formed by joining which of the following sequences? Most of you are giving the correct answer. Yes. So most of you have given this answer, that V, D and J gene segments. You are right. This is what I was actually talking about from the beginning of the class, that CDR3 is encoded by 3 of these segments. Okay, whereas CDR1 and CDR2 are only encoded by V gene segment. Whereas CDR3 is encoded by V, D and J gene segments. So that is the correct solution. Alright, let's move to the next question. P nucleotides are added to the gaps by which of the following enzyme? One interesting thing is happening here actually. So most of your answer are wrong for the first time. Okay, all of you, like most of you have answered the terminal deoxynucleotidyl transferase, but do you want to answer again? I'm telling you that this is not the correct answer. What do you think will be actually the correct answer? D? No. D is not correct. Ligase is an enzyme which joins the phosphodiester bond. Right? Ligase is not an enzyme which can add nucleotides. C, RAG enzymes is also not correct, because RAG is the enzyme responsible for recombination and not for nucleotide addition. So the only answer that remains is obviously B, DNA polymerase. And so because most of your answers were wrong, let's just go through once again this part. Here you would see that these RAG complex creates the DNA hairpin, and the Artemis DNA PK complex, it actually opens up the DNA hairpin. How does it do? So it breaks the it makes a cleavage at the single strand, at one of the strands, like this, here in this particular strand or here in this particular strand. It makes a cleavage, and because of which the DNA hairpin opens up. Okay, and these are actually palindromic nucleotides, right? And then finally, how are these palindromic nucleotides joined? Like, how is this gap, the gap which is generated later on, how is this gap filled? So most of you were answering TDT, but please remember TDT, or terminal deoxynucleotide transferase, adds N nucleotides. So the random nucleotide addition which takes place after these palindromic nucleotides are added, that is actually done by TDT. So TDT adds N nucleotides which are not template coded. But after this, after the pairing of the strands and after these unpaired nucleotides are removed, finally the gaps are filled by which enzyme? DNA polymerase. Right? So here you can see these P nucleotides are these gaps in the P nucleotide are filled by which enzyme? DNA polymerase. These P nucleotides are also filled in. So this gap, this particular gap will be filled in by, here this gap is filled in by which enzyme? DNA polymerase, right? So that is why the correct answer here is the DNA polymerase. The P nucleotide, the gaps in the P nucleotides are filled by DNA polymerase, whereas the N nucleotides are added by TDT. Okay, is that clear with all of you? Please ask me if there is still any other question. Okay. The coding joint and the signal joint. So I did not actually go through the recombination process in detail, but due to the lack of time actually. But in this recombination process, what actually happens is that the V, suppose the recombination is taking place between the V and the J segments, right? Suppose V1 and J2, and when this kind of, so what happens first? The V1 has 123 RSS, suppose, and the J1 has, or the J2 has a 12 RSS, right? And when they are recombining, there are two joints which will be formed. One joint is the joining of the V1 and J2 segments, right, which is the signal joint. And when the RSS they will also join together, because in case of recombination event, that is how it takes place. So when these is, oh sorry, very sorry, this is actually the coding joint, so which will actually code for the VJ segment in the antibody, and this is actually the signal joint. So this you will learn when you actually go through the recombination event in a bit detail, and that is also actually explained by the professor in the lectures. If you follow the lectures, you will understand it better. So this is the coding joint and this is the signal joint, right, which actually are observed during this recombination event. And the coding joint will actually finally code for the antibody light chain, the variable region of the light chain, whereas this signal joint will not code for anything, because this is just a joining of the two RSS sequences, right? Am I clear? Okay, so let's move on to the next question then. The multimeric form of IgM was run on an SDS page under reducing condition. How many protein bands will be observed in the gel? So SDS page under reducing condition means denaturing condition, right? So the protein will be denatured in this case. What do you think will be the answer? How many bands will be observed? So many of you are saying D, 5 bands will be observed. Can any one of you explain why do you think 5 bands will be observed? Some of you were also saying 3 bands will be observed. Can anybody explain? So the correct answer is yes, 2 after the hinge identical site, right? Exactly. So the correct answer is 3. So 3 bands will be observed. How many chains are there in an IgM? There will be 2 light chains and the 2 heavy chains, right? And when we run the multimeric form of IgM on the SDS page, 3 bands will be observed on the gel. Okay, moving on to the last question of today's session. Immunoglobulin chain V gene segment is preceded by an exon which encodes the leader peptide. What is the function of this leader peptide? Yes, most of you are giving correct answers. That is to direct the protein into the cell's secretory pathways. Some of you answered actually that to direct the protein into the cell's nucleus. No, that is not correct because the antibody will actually be secreted. So it will go outside the cell. For this secretion of the antibody, it needs to follow the secretory pathways of the cell, which is through the ER, then through the Golgi vesicles, right? So to direct the protein into the cell secretory pathways, this is the function of the leader peptide. So that is the correct answer. All right.
Surasree,
Yes.
Just one question.
Yes.
This is not relevant to the course. Like, I need a bit of suggestions for the career, like career suggestions. So this is not the right forum to discuss. So is there any way that I can connect to you?
Sure, sure. Um, I will share my, like I had shared the email once, so you can from there you can get my email ID, or here also I'm writing it.
Yeah. Please.
You can connect me anytime.
Okay. Okay. Thank you.
All right, all right. So any of you having any other questions? The recordings will all be there in the YouTube channel. So I will actually share the link of the YouTube channel as well once again in the forum. Book name is Genway Immunobiology. You can also follow Kubi by Immunology. These are the two most followed books of immunology.
Hello, ma'am.
Yes. Please go.
Ma'am, the YouTube link to send it, ma'am.
Um, sorry, I am not actually getting your question.
Ma'am?
Sorry, I did not get you. Can you please tell me in the chat?
YouTube links, ma'am.
YouTube links, I will share actually all these things in the forum. You will get it.
Mail ID, ma'am. Mail.
Yes? Okay, some of you were asking actually the IgM question, right? Okay, so how many light chains are there in the IgM? 2 light chains and 2 heavy chains. Right? Anybody, if any of you having any question related to the course, you can actually post it in the forum. I check the forum continuously, so if you have any questions you can post it there also. I'll get it and I'll discuss it in the class if you have anything related to the course, or if not related to the course, for any personal solution you can actually connect on my Gmail also. So the IgM has 2 light chains and 2 heavy chains. So when they will be actually run on the SDS page, and then they are connected by the, so this is how it looks like, right? So when they will be run on the SDS page, they will form 3 bands. All right? Any other question left? Or else I'll end today's session. Thank you all for joining. We will meet again next time. in this class next Tuesday, 7 to 8 p.m. Alright? Thank you so much for joining. See you again in the next week. Thank you all. Thank you.